going to handle respiration, and specifically cellular respiration. And what you're going to go through are these. Under our respiration, I've tried to summarize it in this form. I'm going to focus mainly on the processes, and you will find out that under respiration we have several processes in which we have glycolysis, Krebs cycle, the electron transport chain, sometimes called the electron transport system. Then we have chemiosmosis. So we have chemiosmosis there, and this one will lead us to phosphorylation. Phosphorylation deals with production of ATP, like that. So we shall look at another phosphorylation in photosynthesis, but that one is called photophosphorylation. It's having that word photo there, meaning light. Then we have this phosphorylation you're going to deal with is oxidative. Then we have another phosphorylation. That one is substrate level. Substrate level phosphorylation. This one is found in the electron transport chain or electron transport system. Then the substrate level, that one is found in glycolysis and Krebs cycle there. So that is it. Those are the kinds of phosphorylations we have. There are three: photo, oxidative phosphorylation, substrate level. But for today, we shall focus mainly on oxidative and substrate level. Then we shall also handle fermentation, and I will look at fermentation in plants and fermentation in animals. So in animals we form lactic acid. Here we shall form lactic acid, and within plants we shall form ethanol. So I'm just giving you a brief preview about what we're going to handle under respiration. I'm going to be focusing mainly on these processes. Then we shall handle some small theory. So I'm also having control of respiration. It's like it's a homeostatic control. There is some kind of feedback mechanisms here. There is something related to feedback mechanisms, and these feedback mechanisms are found in homeostatic controls. So this one relates to homeostasis there. Then we shall end with metabolism of other substrates. So we very well know that under the substrates that are broken down during respiration, This one is dominant, the glucose. Glucose is no, but okay. Glucose will stand in for our carbohydrates as a representative, but we also have fats, we have proteins, and we have to look at how all these are going to be broken down and how they link up with our glycolysis. We shall see how they link up with glycolysis, how they link up with Krebs and the electron transport system. So we shall see the biology there. Then your task will be reading. Try to understand these processes. You read them, write, practice. You do everything. If you can cram, you cram. You find out that when you cram for 3 times, you start understanding. It turns from cram work to understanding. Though cram work is not allowed, but sometimes desperate moves have to be applied if you're desperate enough to understand. So that is it. I will begin with glycolysis here. So under glycolysis, we are having our substrate is going to be glucose. So our substrate is going to be glucose. And from our chemicals of life, I'm going to be relating these things to chemicals of life. So our substrate is this glucose here. I'm going to use alpha glucose. So it's going to be OH CH2 OH. Let me try to reduce this. So this is H, OH there, H here, OH, H. Then I'm having OH and H there. So this is H. I'm going to use my example there. That one is our structure of glucose. Which glucose? Alpha glucose. So you know very well that beta glucose is found in cellulose, and for us human beings and some other organisms are lacking cellulase that is going to break down this cellulose. So it's better we look at that one, alpha glucose. So our alpha glucose is going to be, so you find out that this alpha glucose is an hexose sugar. Hexose sugar from our chemicals of life. It means that This glucose has a hexo, means has 6 carbon atoms. So after knowing that, I'm moving on to our structure here. This is what you have to fight with until you master the steps and the enzymes there. So we shall go through step by step. So I'm having my glucose here. We've seen that it's a 6 carbon sugar. So from glucose, we have to, it's like we pump in ATP. So as we pump in ATP, it's going to be phosphorylated to make it more reactive. So this glucose at this point is going to be, so glucose is phosphorylated here. To make it more reactive. So that's what is happening there. So as we make it more reactive, it's going to turn to a glucose 6-phosphate. So we form glucose 6-phosphate. So when you look at the structure of ATP, the structure of our ATP, let me use a simple structure of ATP, which is the ribose sugar there and the nitrogenous base, then 3 phosphate groups. So it's this phosphate that is going to be broken off. As that phosphate is broken off, it's going to be added to the glucose. So this one is going to be taken by glucose to form glucose 6-phosphate. Are you seeing that? So glucose 6-phosphate. From our simple structure of ATP here, you observe that. So this glucose 6-phosphate is going to be, so now this glucose 6-phosphate is reactive. It was our aim, such that we give glucose a phosphate and make it more reactive. So this reactive glucose 6-phosphate is going to reorganize itself and form fructose phosphate. So it's going to turn to its isomer. I forgot to talk about this. This process is going to be catalyzed by hexokinase enzyme. So it's a hexokinase. How are we going to master the labeling? Hexo, because glucose is a 6 carbon compound. Then kinase, all enzymes that have that kinase part are enzymes that are dealing with ATP breakdown. So kinase, we shall refer that to ATP ATP breakdown. So that is our kinase there. Then we are saying that glucose 6-phosphate turns to its isomer. So we are saying that it's reorganized to its isomer, fructose 6-phosphate. So I'm saying here reorganization to the isomer, which is fructose phosphate, which is fructose 6-phosphate. Now, after that reorganization, I don't know whether this red color is going to be clear. I don't know whether it's going to be clear. Let me turn it to black. It's better I turn it to black. So better I turn this color to black like that. I think now it's clear and visible. So reorganization to its isomer, fructose 6-phosphate. Now, after forming our fructose 6-phosphate, it's going to, remember, these ones are 6 carbon compounds. You will get this in after observing that we have 1, 2, 3, and 4 in glycolysis. Just to get it that we have 4 steps having 6 carbon compounds. So it's going to be easy. So we have 4 steps with 6 carbon compounds and 4 steps with 3 carbon compounds. We shall observe that as we move on. So, which enzyme is responsible for this? It is phosphoglucoisomerase. Meaning, phosphor stands for the phosphate, there. So let me try to do this. Phosphor stands for the phosphate. Gluco, it's this glucose there. Then isomerase, that isomerase part, it means that this enzyme is going to be catalyzing the change of this isomer to that isomer. So this is what the isomerase means there. I hope it's coming out. Phosphoglucoisomerase. Gluco because it's dealing with glucose. And it's turning to its isomer. Which isomer? Fructose 6-phosphate, a 6 carbon compound. Then, when you move on to, so you find out that at least this one is relatively stable. So I'm going to say this one is relatively stable. And you find out that stable compounds are not reactive. It's only after you're unstable that you turn to a reactive compound. That's why we had to pump an inorganic phosphate into glucose to make it reactive. But when it became reactive here to glucose 6-phosphate, it turned to a more stable compound, fructose 6-phosphate. So to turn it to make it more reactive, we pump in another ATP. So this one is to make it more reactive. So when you make it more reactive there, it's going to turn to fructose 1,6-bisphosphate. That one. So we have from our organic chemistry, one phosphate group is sitting at carbon atom 6 and the other one is sitting at carbon atom 1. So it's the reason as to why you have 1,6 and bi means 2 bisphosphate there. So when you look at that, we move on to the enzyme. The enzyme responsible is phosphor because of the phosphate. It's phosphor because of the phosphate. So I'm having phosphor because of this phosphate. Then fructose dealing with fructose. Then kinase. We said earlier on that all enzymes that are having that kinase part are dealing with ATP breakdown into ADP. So when you see that, you'll find out that we formed our fructose 1,6-phosphate bisphosphate. Then from our 1,6-bisphosphate, we have this enzyme aldolase. What aldolase does is it comes to break down this fructose 1,6-bisphosphate, which is now reactive. So just know that this now is reactive. And now as it becomes reactive and unstable, it breaks down into two compounds. We have dihydroacetone. This one is a ketone. Dihydroacetone, which is now a ketone, and phosphoglyceraldehyde, which is pigal. We find this pigal in chemicals of life. The same with dihydroacetone. This one found in chemicals of life, and this pigal is going to be also found in photosynthesis. So I'm going to try to illustrate them to draw their structures. So this is it. I'm having glyceraldehyde here and dihydroxyacetone there. So there are 3 carbon compounds. We saw this in chemicals of life, and due to the presence of this one qualifies to be an aldehyde. So this one's an aldehyde because of that function group being at the terminal and having hydrogen here. Then ketones are always having this keto group there. So I'll call this one aldehyde group and the keto group there. That's why it's called dihydroxyacetone. That one. So these 2 molecules are isomers, and and we are finding them right here, dihydroxyacetone and phosphoglycerolide. So that phosphor is just added on the glyceraldehyde there. We remove one OH and add the phosphate to make it phosphor. So as we do that, I'm moving on to the fact that dihydroxyacetone being a ketone, and we very well know that ketones can't be oxidized. These ones, a ketone cannot be oxidized. These ones are not oxidized. Meanwhile, aldehydes can be oxidized, and you realize that whatever we're going to handle under Krebs cycle, under glycolysis, is a series of reduction and oxidation. It means that if my glyceraldehyde can't be oxidized, yet the steps down here need oxidation, they are redox reactions. It means that my glyceraldehyde has stopped there. It's now having no biological importance. So what we do, we have to convert the ketone to an aldehyde. And as we turn it to an aldehyde, it's going to start doing the work. Are you seeing that? So the body has to turn the dihydroxyacetone to phosphoglyceraldehyde. Then we continue. So it means that I'm having two molecules of with three carbon atoms. We saw that this one is a 3 carbon compound and this one is a 3 carbon compound. So I'm having two molecules, one which was turned by which was converted from dihydroxyacetone and to phosphoglyceraldehyde, and that phosphoglyceraldehyde that was already existing. So I hope you're getting that. Then after, we have to pump in an inorganic phosphate. As we push in an inorganic phosphate, this inorganic phosphate is going to react with the phosphoglyceraldehyde, and we lose 2 hydrogen atoms from each 3 carbon atom. So if I'm having one phosphoglyceraldehyde giving me 2 hydrogens and the other one giving me 2 hydrogens, it means that they are going to give me 4 hydrogens. It's what I've indicated here as 2 times that. So, but when we give off that, when we give in 2 hydrogens, these 2 hydrogens are picked up by NAD, oxidized NAD, and it's going to be turned to reduced NAD like that. Now as it turns to reduced NAD, every NAD atom, NAD molecule, gives us 6, it gives us 3 ATPs. This one is found, this analysis is in the electron transport system. It means that if another NAD oxidized NAD comes for that hydrogen there. It's going to be turned to NAD, reduced NAD, and gives us 3 ATP. So this is in the electron transport system. It means that the overall is 6 ATP produced. Are you seeing that? Just from glycolysis at this point. But remember, we spent some ATP up here. So this ATP, one molecule was used up there, and one molecule was used up there. So I will circle them like that, and this. So we've used up to, we've manufactured 6. So we move on. Now, as this process takes place, we are going to end up forming, glycerate. We end up forming glycerate 1,3-bisphosphate. So this glycerate 1,3-bisphosphate is going to be turned to glycerate 3-phosphate. So what happens is that this from our piggyl here, we had one phosphate group. We had one phosphate group that was attached to the glyceraldehyde molecule. Then we added the second one, so there are 2, and one is attached to carbon atom 1 and one attached to carbon atom 3. That's why we call it glycerate 1,3-bisphosphate. But now we'll bring in our ADP, and it's turned to ATP. As it's turned to ATP, it's going to pick that inorganic phosphate that was at carbon atom 1. So we end up with glycerate 3-phosphate. It means that the one that was at carbon atom 1 is now lost. To ATP, like that. So as we form glycerate 3-phosphate, this glycerate 3-phosphate is going to be used to form ATP and loses water, and you just understand that as we bring in our ADP here, that ADP is going to take the last phosphate that was at carbon atom 3, and we end up with pyruvate. And when you look at the structure of pyruvate, which is CH3, COOH, pyruvic acid or pyruvate. It has 3 carbon atoms. It has carbon atom 1, it has carbon atom 2, it has carbon atom 3. There is no phosphate. It has no phosphate group. So after seeing that, I want you to observe keenly this. We have 2 molecules of our pyruvate, 2 molecules of our glycerate 3-phosphate, 2 molecules of glycerate 1,3-bisphosphate, and 2 molecules of phosphoglyceraldehyde. So how did it come about? It came from this ketone there that was turned to that molecule. So whatever we were forming here, you find out that I formed 2 ATP, and this ATP is formed by substrate level. So are you seeing where the substrate level phosphorylation? Wherever we form ATP, that is phosphorylation that is forming that ATP, the process. And now this one is substrate level phosphorylation. So that is substrate level phosphorylation. And this ATP here formed is also as a result of substrate level phosphorylation, like that. And the ATP that is formed here, these 6, these ones as a result of the electron transport system. And we know very well that this one is oxidative. Oxidative phosphorylation, like that. So that is glycolysis. See, the aim of glycolysis is formation of that pyruvate there. So try to understand this, try to memorize it. It will help you somewhere. It will help you. It will bring a scenario that needs this understanding. So I'm going to move on to Krebs cycle. So after our formation of by the way, location, we didn't talk about location. So location, so this process is within the cytoplasm, and so this process is within the cytoplasm. Then what else should we add on? Significance of glycolysis. Significance of glycolysis. So significance of this glycolysis, we have 1. Let me change this to blue. So significance of glycolysis, we have the first one. The first one is ATP for powering cellular activity. So this glycolysis is a source of ATP. Source of ATP for powering cellular activities such as cell divisions. Are you seeing that? Then the other significance is production of NAD, of reduced NAD. So it's a source of NAD H2, and this one provides more energy for electron transport chain. So this NAD is carrying electrons, and these electrons are needed for the electron transport system. So source of NAD for the electron transport system. Then 3 is forms pyruvate. So under glycolysis, our intentions are to form pyruvate. So pyruvate is the same as pyruvic acid. So we are forming pyruvate, which is a substrate, for the Krebs cycle. So that is it. Now we have to count the ATP expenditure. So our ATP expenditure and production. So from our analysis, we spent 1 and 2. So we spent 2 ATPs, and we produced, if you remember, we produced 1 and 2 ATPs. Those ones were by substrate level. So we produced 2 ATPs by substrate level. Phosphorylation. Then we produced more 6 ATPs by the electron transport chain. So this one was oxidative. By oxidative level phosphorylation. So you find out that our net production is 6. So net ATP count is 6 ATPs. Why? It's because these 2 are going to be used up. We always use 2 ATPs, and there are these ATPs that are formed here. So what we remain with is this. So after knowing that, we move on to glycolysis, to Krebs cycle. But before we even reach that Krebs cycle, there is a process that is very important to look through. There is an important process to look through, and that's the process that happens before we enter the mitochondria. It's this process here, that conversion there, before we reach the mitochondria. So what happens is that during the Krebs cycle, or before, let me say before, let me put here, before the Krebs cycle, I'm having so the location, so our location is going to be the inter membrane space, or I will call it that process when, remember, our mitochondria is double-walled or double membraned. So that process when you're moving from the cytoplasm to the mitochondria matrix. As you move from the cytoplasm to the matrix, what happens is that we have to first convert our pyruvic acid, or the pyruvate. We have to first convert it to acetyl coenzyme A. So let me bring this close. I don't want to repeat stuff. So we are having a change of our pyruvate. We saw the structure, it's this, turning to acetyl coenzyme A. So what happens is that we are having pyruvate dehydrogenase enzyme at this point here. We have pyruvate dehydrogenase. Dehydrogenase enzyme. And what it does, it's going to catalyze these conversions. So from pyruvic acid to we are going to first decarboxylate it. So we remove carbon dioxide. As we remove the carbon dioxide, just imagine that we've removed this part here. We are going to remove this carbon dioxide at that point. So as we remove that carbon dioxide, we remain with hydrogen. And this hydrogen, if you took in enough, you have to remember that we formed the two pyruvate molecules from glycolysis there. We formed the two pyruvate molecules. It means that this hydrogen is going to be, we have two hydrogen molecules and 2 carbon dioxide molecules are going off there. So these two are going to be taken by NAD. So our NAD takes those hydrogens there. As it takes the hydrogens, the whole process is catalyzed by pyruvate dehydrogenase enzyme, and after we've remained with only this. We've only remained with that acetyl part. So what do? We come and fetch the coenzyme A and add it there. So hope that one is clear. That is the process. If you master, or if you understand the structure of pyruvic acid, you can at least narrate the process there. So after forming our coenzyme A, acetyl coenzyme A, let me write it here. Acetyl coenzyme A, or sometimes called acetyl co A. All these ones are accepted. So we have that. Then I'm going to move on with my 2 carbon acetyl coenzyme A. Remember, it has 2 carbon atoms, carbon atom 1 and 2. The third was taken. So I'm moving on with my 2 my coenzyme A, acetyl here, and I'm now going to pump it into the Krebs cycle. But what we have to realize is that the location, so our location it's the mitochondrial matrix. Mitochondrial matrix, that fluid within the mitochondria. So that's where specifically Krebs cycle is going to be. So under this, we are going to insert there our coenzyme A, 2 carbon atoms. Now this one's going to combine with oxaloacetic acid. It's going to combine with oxaloacetic acid, and it will end up giving us citric acid. So oxaloacetic acid is a 4 carbon compound. As this 4 carbon compound joins with a 2 carbon compound, they will give us a 6 carbon compound there. So this is citric acid. Some books say citrate, but it's not. The two are the same. Citric acid, they mean the same thing. So after that, we are going to give off carbon dioxide, and we give off 2 hydrogen atoms for every molecule of acetyl coenzyme A. So when I multiply by 2 here, it means that now there are 2 molecules, and each is giving me 2 hydrogens. which is giving me 2 hydrogens. Why? Because we fed into pyruvate. 2 pyruvate. This 2 pyruvate gave us 2 acetyl coenzyme A. So I think that one is clear. So let us not confuse them. So after that, these processes are very long and we can't exhaust all the enzymes that are going to be active here. But what we have to know, at least these are the basics: loss of carbon dioxide, formation of 2 pairs of hydrogen atoms. So these ones are going to be picked up by NAD. So these ones are picked up by NAD and forming NAD, reduced NAD, like that. So if there are 2, remember NAD can only take 2 hydrogens. So if there are 4, we need 2 NAD molecules. And every NAD molecule gives us 3 ATP. And we have 2 NAD molecules, so 3 ATPs. So when you combine this, they give you the 6 that you're seeing there. So that is it. After that, we have alpha ketoglutarate. So this one was a 6 carbon compound, citric acid. As it lost this carbon dioxide, we formed the 5 carbon compound, alpha ketoglutarate. Alpha ketoglutarate is also going to lose the carbon dioxide there. So I'm going to enclose my alpha ketoglutarate like this. Or is it not reacting? Oh, let us leave it. Okay, we form carbon dioxide, and the carbon dioxide that has gone, remember now there are 2 molecules. At this step we have loss of 4 hydrogen atoms that are going to yield 6 ATP molecules. Now these 6 are going to be carried by they are going to be as a result of 2 reduced NAD molecules. Then we have this ATP, these 2 ATP molecules. These ones are directly from substrate level. So this one is a direct production of substrate level. Level phosphorylation. So from this, we have more too. There is production of GTP. So GTP, 2 molecules of GTP are going to be turned into 2 molecules of ATP. This ATP is also going to be counted there. So from that we have succinate or succinic acid. So after forming our succinic acid, which is a 4 carbon compound, this succinic acid is going to lose 2 hydrogens. But remember that those 2 hydrogens, we have to multiply them times 2 to get the overall number there. So we have FAD. FAD is going to come in at this step. Hydrogen, we saw that hydrogen is usually fetched by NAD. So oxidized NAD comes for the hydrogen and takes to the electron transport system to form ATP. But for this case, it's different. It's going to be NAD just after succinic acid or succinate. We have NAD coming for these hydrogens. As NAD fetches those hydrogens, we are forming two molecules. So we have two NAD, oxidized NAD. We form two molecules of reduced NAD. And those two molecules are going to give us four ATPs. Why? Because NAD, for every NAD molecule, just keep this in mind that for every FAD molecule, we produce 2 ATP. And for every NAD, we produce 3 ATP, like that. So we move on to, we have addition of water there. So we have to pump in some water. As we pump in some water, we form malate. And from malate, we have to lose 2 hydrogens. And these 2 hydrogens are going to be from 2 molecules. So we form 3 ATP molecules, 6 ATP. So after that, we form oxaloacetic acid. We shall call this oxaloacetic acid, and the cycle continues. That's why it's called a Krebs cycle. So oxalo goes back and fetches more acetyl coenzymes, coenzyme A forming citric acid, alpha-ketoglutarate, succinate, malate, and oxaloacetic acid. So that is it for Krebs cycle. So we have to talk about the importance of Krebs cycle, and we have to count the number of ATPs that are formed here. So let us first give an account of the number. So ATP count, I will call it ATP count. So it's going to be, we have so let us count them together. We have 6 ATP molecules there. 6 ATP. When we add on 6, that's 12. When we add this 6, that's 18. So when you add on this 4, that's 22. Then when we add on this ATP here, that ATP there, it will give us 24. and that one there gives us 26. Then from our 26, when we add on what else we added 18, 22, 26. Yeah, I think that's it. So, but remember, okay, yeah, that is it, 22. Then when we add on 26, when we add on the 6, oh yeah, this is it. We had forgotten we had left out this count here. Remember, this NAD is coming from one pyruvate molecule. So if there are 2 pyruvate molecules, this NAD gives us 2 NAD reduced NAD. So if there are 2, they end up giving us 6 ATP molecules. So when we add on the what? So when we add on the 26 plus that 6, then remember that in glycolysis we came up with a net count of 6. So we had a net count of 6, so I'm going to add that 6 there. So this one gives us 30, gives us 32. Then 32 plus the 6 from glycolysis, we end up with 38. So 38 ATP in total. So that is it. So I want to talk about the importance of Krebs cycle, if maybe it's asked. Importance of the Krebs cycle. Or its significance in life. So under this we have 1, it brings about the generation of macromolecules. Brings about the generation of macromolecules. So it brings about breakdown of large molecules into smaller molecules. That's catabolism. Then it provides the reducing power for electron transport system. So provides the reducing power for the electron transport system. What do I mean here? I mean that it provides the electrons this reducing power is the electrons that it's going to be giving through. It's going to be providing electrons through our node. This one's a carrier. Then our other importance is it's an interconversion center. So it's an interconversion center. So what do I mean? It's a source of intermediate compounds for the synthesis of fatty acids, amino acids, carbohydrates. It's a source. It's a source of intermediate compounds. For synthesis of fatty acids, amino acids, and carbohydrates. I think that. So it's also a source of hydrogen ions. It lies within the reducing power source. Source of hydrogen ions. So how is it a source of these hydrogen ions? Is through the very many NAD molecules that it was forming and fed. So those are the ones that are going to be moving hydrogen ions from Krebs cycle to the electron transport chain. Then we have to talk about the roles. You may be asked to state the roles of coenzyme A. So the role of coenzyme A. Yeah, and one is it activates acetate, or it activates our activates acetate. Then so the acetate I'm talking about is this this group here, that group there. So if it's not there, then that acetate is not active. So that is it. Activates that acetate, then transfers acetyl group to form citrate. So transfers transfers the acetyl group to oxaloacetic acid to form citric acid. Then the other role is acts as a central link for proteins, fatty acids in the Krebs cycle. So acts as a central link for proteins, fatty acids, and into the Krebs cycle. Then we have to talk about the similarities between glycolysis and the Krebs cycle. So similarities between glycolysis and our Krebs cycle. And within this, we shall cover that in both glycolysis and Krebs cycle, we have NAD, reduced NAD, ATP are formed. So in both NAD, reduced NAD and ATP are formed. Then the other observation is these ones are both occurring in living cells. Occur in living cells. So you find out more similarities. Find out more. What I'm going to talk about next are the differences and we shall see differences, and I'm having glycolysis and Krebs there. So the first difference is that fat is not involved during glycolysis. You've observed that fat is not involved. But again, this way, where in Krebs we have fat involved. Then we have carbon dioxide this way. So carbon dioxide not formed, and this way in under Krebs cycle we have carbon dioxide formed. So the processes there that you have to recall that we're forming carbon dioxide at that point and at this other point there we formed carbon dioxide. So the other difference we can talk about is this one, the location occurs, glycolysis occurs on the cytoplasm, and the Krebs cycle occurs in the mitochondrial matrix. Occurs in the mitochondrial matrix. Then it doesn't require oxygen to occur, so no oxygen is required. But under Krebs cycle we need that oxygen. Why? This oxygen is an electron acceptor in the electron transport system at the end of it. So if this oxygen is absent, the whole process is going to stop right from the Krebs cycle, the electron transport chain. So we need that oxygen. Then glycolysis doesn't involve coenzyme A. Doesn't involve coenzyme A. But our Krebs involves coenzyme A, like that. So these are the differences. Then we move on to the electron transport system. So under our electron transport system, I'm having my illustration here for the electron transport system. This one summarizes every step that is needed to be talked about. It's a series. You have to first know that this pathway is a series of redox and redox reactions, reduction and oxidation. So I'll move straight to the content. So after our NAD, after this NAD, picking, after that NAD picking, after manufacturing our NAD or formation of NAD within the Krebs cycle, this NAD moves straight to the electron transport system. And within this electron transfer system, what we have to know, the location. First, identify the location. So it's located within within the inner membrane. Which membrane? The mitochondrial membrane. Mitochondrial membrane there. So what happens is that we are going to our node is going to move from our glycolysis, because we have some production of NAD, reduced NAD there. So when I say oxidized NAD, I mean this NAD with a positive charge. Reduced, we know very well that reduction is addition of hydrogen. So by the way, those are the terminologies we have to first look into. So we have reduction and oxidation. So when we say that a certain molecule is going to be reduced, we mean it's going to be added. We mean there is an addition of hydrogen atoms or removal of electrons. Is it removal of oxygen? Okay, of oxygen atoms. Then lastly, there is addition of electrons. Let me give it space. So addition of electrons. So those are all definitions of reduction. Then for oxidation, we can have I'm going to follow that criteria. We'll have removal of hydrogen atoms. We say that molecule has been oxidized due to removal of hydrogen atoms. Then addition of oxygen, then removal or loss of electrons. So it can also be loss of electrons. So these are the series of reduction and oxidations that are going to be taking place. First, reduced NAD submits in the hydrogen and the electrons it's carrying. So as it submits in the electrons, it's going to give them to the first protein complex that we have. This one is also a protein complex. We call it protein complex 1. I'll call it complex 1. So within this protein complex, I'm having, maybe let us first go through the structure. I'm having FMN, which is our flavin mononucleotide. Flavin mononucleotide, FMN. N for nucleotide, M for mono, and F for flavin, like that. So, and this one is called an iron sulfur protein. So we have an iron sulfur protein there. Then we have coenzyme Q at that point. This one is coenzyme Q. Then I'm having protein complex 2. That is our protein complex 2. It's slightly above that coenzyme Q. And for it, it's having FAD there. It has FAD there. Then I'm having another iron sulfur protein. Then I'm having our protein complex 2, protein complex 3, and within this protein complex 3, I'm having 3 proteins. I'm having cytochrome B, iron sulfur protein, and cytochrome C. So this one is cytochrome B, and C, cytochrome A, all that stuff is there. Then we move on to protein complex 4. And within this protein complex 4, we have cytochrome A and cytochrome A3. Are you saying it's cytochrome A3 that's going to submit the electrons? And the 2 of them, these 2 are going to be taken by the hydrogen later on. It reacts with the oxygen and give us water. So I'm going to try to balance this. Oxygen with the 2.5. So what really happens is that when you're trying to explain this, you will start by saying that during the electron transport process, electron carriers alternate between reduced and oxidized states. So we shall say electron carriers alternate between reduced and oxidized states, and they donate electrons. So let me try to explain it from this very drawing here. So this NAD is an electron carrier. It's going to give our FMN. Now as it gives the electrons to FMN, these two electrons, it's going to be oxidized. As it's getting oxidized, it leaves this FMN reduced. It leaves it in a reduced state. Why? Because this one has gained the electrons that were within the reduced form. So now this one has gained electrons. What do we call gain of electrons? That is reduction. So our flavin mononucleotide has been reduced. So what flavin mononucleotide does, it submits those electrons to the iron sulfur protein. So as flavin gives in, it's losing the electrons. That's what we call oxidation. So as it gets oxidized by losing the electrons, it's going to reduce the iron sulfur protein. So as the iron sulfur protein gets reduced through gain of electrons, it's reduced. So as it gets reduced, it's going to be forced to give in these electrons to our coenzyme Q. Now as our coenzyme Q picks up these electrons, it's going to get reduced. Meanwhile, as it's getting reduced, the one that had the electrons before is now getting oxidized. Now, coenzyme Q submits these electrons to cytochrome B. Now we've moved to our protein complex 3. So electrons are given to our cytochrome B. Cytochrome B picks up these electrons and submits them to iron sulfur protein. Now it's this iron sulfur protein that's going to submit the electrons to cytochrome C. Are you seeing that? So that process is a chain. It's a chain process where we have gain and loss, gain and loss. Who gains electrons is that species that is going to be reduced. If you gain electrons, you get reduced. If you lose them, you get oxidized. So at this point, our cytochrome C has gained. But now, if we want to reduce it, we make it lose those electrons. So it loses the electrons and it gets oxidized. So at that point, we have loss. Now, as it gets oxidized, it's going to give cytochrome A in protein complex 4. It's going to give him, or to make him gain, cytochrome A has now gained the electrons. So cytochrome A is now reduced. As cytochrome A submits those electrons to cytochrome A3, we shall say cytochrome A3 has gained. Therefore, cytochrome A3 is now reduced. So, as it's now reduced, what we do, this cytochrome is going to lose those electrons and we shall say that it's getting oxidized. But there is an enzyme that is recognized here. That enzyme is cytochrome A, oxidase enzyme. So it's cytochrome A oxidase. I'll call it cytochrome oxidase. So that cytochrome oxidase enzyme is going to catalyze the oxidation. So it's going to catalyze the oxidation of, or catalyze the loss of the electrons. So it's going to catalyze the loss or the oxidation of electrons from cytochrome A to the oxygen. It's this process that is occurring here. There is a loss of electrons there. Now, as we lose these electrons, this is the reason as to why we say oxygen is an electron. The last is the last electron acceptor. Why? Oxygen will come and pick up those electrons and form water. This is now the metabolic water they usually talk about. So this is now our metabolic water there. So that's the process. What we have to first master is the structure and what happens. So when you master that and the proteins that are engaging in this process, you find out that things are now going to be moving as planned. So I'm going to talk about this complex 2. We saw that NAD submits its electrons at complex 1, but now FAD submits its electrons at complex 2, and as it submits these electrons at complex 2, they are going to be picked up by FAD, meaning FAD is going to be reduced. As FAD submits the electrons to our iron sulfur protein, it's going to be oxidized as sulfur, and sulfur protein gets reduced. And the iron sulfur protein will finally submit these 2 electrons to coenzyme Q. Coenzyme Q follows a similar path. Now, I made this in this kind of slanting form. Why? Because electrons that are carried by NAD are going to be electrons of higher energy, at higher energy levels. So these ones are at, let me say, very high energy level, such that when these electrons are submitted, they are going to be moving from regions of higher energy coming down to the ground state. Now this energy that is contained in these electrons is going to be tapped. That's why we have to study that chemiosmosis. Why? There is a process that is occurring within here. There is a process occurring here, and that process is proton pumping. So take this as the mitochondrial matrix, and this side the intermembrane space. That space between the 2. Did I illustrate it here? Okay. That space between the 2 membranes, if I'm having this as the first membrane of the mitochondria, then the second membrane, so this space, that space there, is the intermembrane space. So this intermembrane space there. So we have hydrogens that are going to be pumped from the matrix to the membrane space. But these hydrogens are going to be pumped depending on this energy that is being tapped off. So as electrons are moving from FAD to our iron sulfur protein, that energy they are carrying is going to be picked up and used to pump hydrogen from the mitochondria matrix to the intermembrane space. And the end result is going to be an increase in concentration of hydrogen ions. So the hydrogen ion concentration increases within the mitochondrial matrix, the mitochondrial space, intermembrane space there. So that process is also occurring here. So we are pumping there and also here. Just know as electrons are moving, that energy is tapped off. That energy is tapped off and used for that business. So what do we say? We are going to find out that this is the reason as to why NAD produces only 2 ATPs. It's the reason as why FAD produces 2 and NAD produces 3 ATPs. I think the point at which NAD is submitting the electrons. So at this point we have hydrogen, influx of hydrogen into the mitochondrial space, intermembrane space. So as these electrons, one electron does that activity and the other electron, the energy possessed by the other electron is going to be also used there and pumping the hydrogen there. So what happens is that we form using NAD. So using NAD, using our reduced NAD, we pump 3 hydrogen atoms into the membrane space. As we pump 3 hydrogen atoms in the membrane space, every hydrogen atom is going to give us 1 ATP. So the total will be 3 ATP molecules. Now, since FAD gave us those electrons at a very low, at a slightly lower energy level, around this one is slightly low. So because FAD submitted them at a lower energy level, it's going to only pump 2 hydrogens. So FAD, the electrons submitted by FAD are going to only pump 2 hydrogens, and every hydrogen ion is responsible for at least 1 ATP molecule. So if there are 2, they are going to give us 2 ATP molecules. So you're free to pause and try to unwind and listen to this audio and the video very well. You note down important areas you need to. And the other task you have is go to those textbooks and read the essay. Go to the textbook, read about how they explain this thing, then I'm going to be providing my summary at the end of this recording. I will take a pic of my summary and put it there. So that is the process. I'm going to maybe, should I rub off this? Should I rub off this? No, let me leave it. Let me just move straight to chemiosmosis. Chemiosmosis. The reason as why I'm not giving my summary, my essays, these essays are, I made them according to the way I understood the thing. So I need you to also find ways of understanding the thing. Then you write down your understanding. You write down something that you, the author, understands. That one will be very important for your email. If there is a paper tomorrow, just go and read the summary. Don't fall into books. You may end up even fainting from books. So I'm moving on to the chemiosmotic theory or chemiosmosis. This one is going to deal specifically with the hydrogen ion pumping. We are seeing the relationship between hydrogen ions, ATP production, and the electron transport system. All those factors have to be resolved here. Factor number 1. So process 1 is the electron transport system and how it links up with the phosphorylation, which is ATP synthesis. Then factor number 3, which is that is it. Okay, hydrogen ion, shall say hydrogen ion pumping, but this one is more into ATP synthesis, phosphorylation. So what happens is that our electron transport system provides, as electrons are moving, they are going to provide that energy that is needed to pump hydrogen ions into the intermembrane space. So this one is just giving us that energy. These electrons are carrying some energy. If you go and study about radioactivity and the hydrogen spectrum in chemistry, you will find out that as electrons jump to a higher energy level, there is that energy they possess. Now, as these electrons are coming back to a lower energy level, this is all chemistry. As they come back to a lower energy level, there is that energy they are going to lose. So they lose that kind of energy. So energy is lost. So this is the same process that occurs within photosynthesis. It occurs within photosynthesis as light strikes the leaf. It's going to cause electrons to move from their original state to higher energy levels. Then as they come back, we shall talk about that electrons within the photosystem, the pigments, what. So as they come back to lower energy levels, there is that energy they are going to give off. So in our biological systems, this energy is not just lost like that. So we have to tap that energy and use it in a useful maybe process, such as pumping of hydrogens. So as we pump hydrogen ions, this is what occurs. This is what happens here. So our NAD as usual. So this NAD is coming from glycolysis and Krebs cycle. So it's going to submit its electrons to FMN in our protein complex 1. Now these electrons are going to be picked up by our iron sulfur protein. But as you're transferring those electrons, they have to lose that kind of energy that we're interested in. So as they lose that energy, moving from high energy level to a lower energy level, we use that energy to pump the first hydrogen ion. So that electron is going to move to coenzyme Q. Now as it moves to coenzyme Q, it's going to proceed. Remember, it's a series of redox and deoxidation. So as coenzyme Q loses the electrons, it's going to become oxidized. Now we have our cytochrome B picking up the electrons. As cytochrome B transfers the electrons to the iron sulfur protein, that very energy that is possessed by those electrons is going to be lost at that point, and we are going to time that very moment and use that energy to form to pump hydrogen from the inside the matrix, from the mitochondrial matrix to the intermembrane space here. So this one is all our intermembrane space. So after that, we wait for that electron to move. It goes to our cytochrome C and it moves to cytochrome A. Now as it moves from cytochrome A to our cytochrome A3, it's going to lose the final energy that we want. So that energy is also used to pump hydrogen from down to up over to the intermembrane space. Now we've pumped 3 hydrogens. This process is going to occur. We don't have the body is having very many NAD molecules. We are not limited to one, as this illustration says. We have very many reduced NAD molecules within the process. Now all these ones are going to participate in pumping hydrogen into the membrane space such that it's going to reach a time and our hydrogen ions concentration increases. So as it increases, remember that FAD is also bringing in. So as FAD also brings in, this concentration is going to increase. Now as our concentration increases, there is a gradient that is going to build up. Remember, take note that this membrane that you're talking about is impermeable to hydrogen ions. So this membrane is very impermeable to hydrogen ions. So the only place that is permeable to hydrogen is through this stocked granule, which is an enzyme, ATP. It's called our ATP synthase. So that one is our ATP synthase enzyme. So hydrogen can only pass through this because of its high concentration. It wants to move to regions of low hydrogen concentration. but it can't do that without passing through the recommended channel. So what it does, it has to go to this channel and start moving. So our hydrogen is going to begin moving from regions of high concentration to regions of low concentration, like this. Now, as one hydrogen molecule passes through the channel, it's going to induce this channel to generate that energy that is needed to combine ADP with an inorganic phosphate and form ATP. Then the other one comes. So 3 hydrogen ions passing through our ATP synthase are going to produce 3 ATP molecules, and that one is a characteristic it's going to be related to NAD, reduced NAD. Then our FAD is going to pump to 1 and 2. So as it pumps that 1 and 2, as they move through this our ATP synthase, are going to bring up only 2 ATP molecules, like that. So that is the chemiosmotic theory and chemiosmosis. That's how we explain it. Try to relate it to the electron transport system, because in actual sense the electron transport system is just used to produce that energy that we shall use to pump hydrogen across the membrane. Then, as the hydrogen comes back, we pick that energy that we want to form ATP. Now this ATP is going to be taken for other use. Example, cell division, protein synthesis, all those processes. Then, I'm having my card definition here. Chemiosmosis is a process in which energy stored in form of hydrogen ion gradient across a membrane is used to drive cellular work, like ATP synthesis. So that is it, chemiosmotic theory. Then we move on to our fermentation. So when we talk of respiration, we have to talk about fermentation. Sorry, it's a must to talk about fermentation, because fermentation is anaerobic respiration. Respiration in absence of oxygen. And during this fermentation, remember we have 2 kinds of fermentations. We have that anaerobic fermentation in animals, call it what? Anaerobic respiration in animals, and the anaerobic respiration in plants. I will begin with that one of animals, just to gain understanding and pass through the process quickly. So under this fermentation, we very well know that it's anaerobic respiration. So this is that fermentation there is anaerobic it's anaerobic respiration. So honey in absence of oxygen. Then we have that occurring in animals. So we have one occurring within animals, and we have that occurring within plants and yeast there. So what happens is that we undergo glycolysis. Our glycolysis doesn't require oxygen. Even in our difference, we stated that glycolysis and Krebs. This glycolysis doesn't require oxygen. So glycolysis is going to take place and we end up with pyruvic acid. Now, this is our pyruvic acid or the pyruvate. The structure we now know. So, because of absence of oxygen, let me take you back to our electron transport system. Because of absence of oxygen, who is going to be available to take the electrons? There is no electron carrier if oxygen is absent. So, just get it that if the electron carrier is not there, electrons are going to block this entire system from happening. That entire system up to where NAD submits the electrons. It's like jam. So electrons are going to cause jam. So electron jam within the system. Now as the system gets jammed up like that, it's going to, let me remove this. So NAD will no longer submit those electrons. Why? Because we have no way of giving in these electrons. No, the whole process is blocked. So as the process is blocked, our NAD carrier, our hydrogen carrier and electron carrier, which is NAD, is well known for that duty. What it does is going to bring these hydrogen ions that it had. So you remember that you have to first understand that NAD, reduced NAD, is of that structure. But if you try to oxidize it or to make it lose the hydrogen, it's going to be oxidized NAD. So, and 2 hydrogen ions. Where are the electrons? We have 2 electrons. So these are the 2 electrons that NAD was carrying and the 2 hydrogens. Are you seeing that? So what happens is that NAD is going to come and submit these 2 hydrogens here. These are the 2 hydrogens from NAD. So when you look at animals, after lack of oxygen, this pyruvic acid is going to be reduced. NAD will look for where to put the hydrogens in the electron transfer system. There is no, then it will come back and fetch this pyruvate. Instead of pyruvate entering the mitochondria, it's going to be given the hydrogens. And from our organic chemistry, we know very well that when we add the hydrogen to such an organic compound, let me try to expand it and you see H3, C, COOH. This acidic part is not going to be reduced as such, but the carbonyl group is going to be reduced by the hydrogen to what? To an alcohol. So let me cut that and put it here. So it's going to, this is what happens. The hydrogens are going to attack that carbonyl group, and this is what happens. The hydrogens that NAD submitted are going to do this. CH3, C. They will break that bond, add one hydrogen there and hydrogen there, then they leave the carbonyl group intact. So we end up with such an acid, which is lactic acid. That's why we usually say that there is lactic acid accumulation during anaerobic respiration in muscles. So this is the lactic acid that has accumulated. So that is what happens if there is no oxygen. First, if there is no oxygen, the electron transport system is going to be blocked by the accumulation of electrons. Then NAD will no longer submit electrons to a blocked rod or a blocked route. What it does, this NAD is going to start moving and looking for, remember, if the electron transport system is blocked, Krebs doesn't function. So the Krebs cycle stops functioning. As for glycolysis, that glycolysis still continues, but what it does, it takes an alternative route. Give the hydrogens to pyruvate. Pyruvate turns to lactic acid. So that is it. Then we move on to fermentation in plants. So under fermentation in plants and yeast, we are seeing that lactic acid is first decarboxylated. Removed, carbon dioxide is first removed. It's removed. We call this one decarboxylation in organic chemistry. So as we remove this carbon dioxide, take this as take it that this part is what we've removed. We remain with what? We remain with, okay, let me illustrate it here. So this one happened in animals. So what about plants and yeast? What happens in plants and yeast is that our pyruvic acid is going to be decarboxylated in this way. This is going to be scrapped off to form carbon dioxide, and we end up with CH3, COH with a double bond there. So what's this? This is ethanol. Are you seeing that? This is ethanol. Ethanol is analdehyde. Now, as we form our ethanol, some books called it acetaldehyde. There is nothing much about that, but the easiest is ethanol, we know that. So from our ethanol, remember that now NAD has nowhere to put these hydrogens except bringing them back to our ethanol. So ethanol is an aldehyde. Aldehydes can be reduced. So we bring these two hydrogen ions and add them to our ethanol. We end up with this. Are you seeing that? So this is ethanol. You can rearrange it in a recognizable state, CH2OH. So this is our ethanol. This is the reason as to why under alcohol fermentation. So this is lactic acid fermentation, and this is alcohol fermentation, alcoholic fermentation. We end up with an alcohol, but usually carbon dioxide is removed from such a process. At least I've explained why that carbon dioxide is always seen there as a lost carbon dioxide molecule. So this is our fermentation. Go and read. At least I've given you a good start for these two fermentations. If you pair it up with organic chemistry, reduction, oxidation, things are going to come out. So I move on to our interconversions of carbohydrates. I'm closing the topic. So we are left with only this control respiration, and that's it. We shall be done. So we move on to metabolism of other substrates. And under this, I'm going to focus on the interconversions of carbohydrates, fats, and proteins. Usually they say when your carbohydrates are done in the body during starvation, the body resorts to fats and proteins for formation of energy. So you may wonder, how would we get energy from proteins? How do we get energy from fats? So from our illustration here, I'm having carbohydrates as the main energy source. So we know very well that this is now glucose, and from our carbohydrate there, one arrow, these arrows are showing that it's a reversible reaction. Reversible reaction. So our carbohydrate, call it the glucose, is going to be phosphorylated into 3 carbon sugar. So it's going to be phosphorylated into a 3 carbon sugar there. What do we mean? This one is bigger. So that one is a 3 carbon sugar, phosphoglyceraldehyde. And this phosphoglyceraldehyde will undergo series of reactions to form pyruvic acid. Now, it's this pyruvic acid that turns to acetyl-CoA. And from acetyl-CoA, we go in for the Krebs cycle there. So, if your carbohydrates are done in the body, what happens? You resort to your fat storage, your adipose tissue. start breaking down the fat into, so you start breaking down your fat into glycerol. We know glycerol is and fatty acids are constituents of fat. So let me begin with glycerol. As you break down the fat, you form glycerol. Then this glycerol, we very well know that it's a 3-carbon compound with 3 OH groups there. This is H2, H2, like that. So this glycerol, if you want clarity on this, go to chemicals of life. Go to chemicals of life. So this glycerol, since it's a 3-carbon compound, it can be easily turned into this 3-carbon sugar phosphoglyceraldehyde. And when you turn glycerol to phosphoglyceraldehyde, the journey continues into pyruvic acid. So there are very many reactions that are going to occur within that place there, and some reactions are not documented. It's only by chance that you fall onto such a reaction. This is it. It's only by chance that you fall onto such a reaction, and usually you'll always get such illustrations after after mock. But for now, I've presented it to you. For me, I got it after mock. I found it in a mock solution bank. So I'm having my glycerol here. And from my glycerol, I want to see how this glycerol is going to be turned into an energy producing molecule. So what we do is that we first phosphorylate it. So it's going to undergo phosphorylation. What do we mean? We are going to pump in ATP into this glycerol. As we pump in ATP, we form glycerol 3-phosphate. Now as we form glycerol 3-phosphate, it's going to be dehydrogenated. What do I mean? Hydrogen is going to be scrapped off from our glycerol. So we very well know that this NAD is responsible for picking hydrogen atoms from our molecules or from organic compounds. So as we dehydrogenate it, forming reduced NAD, we are going to form dihydroxyacetone. You remember that isomer of pigar. So this dihydroxyacetone is a ketone. We can't oxidize ketones, and they are relatively stable to attack. So we have to isomerize it to pigar phosphoglyceraldehyde. As we isomerize it to pigar, we continue with those normal steps that we had: pumping pump in an inorganic phosphate and remove two hydrogens. This is the hydrogenation. You form glycerate 1,3-bisphosphate. From glycerate 1,3-bisphosphate, you start harvesting your ATP. You form glycerate 3-phosphate. Now, from our illustrations that we had before, I jumped from glycerate 3-phosphate straight to pyruvate. Books always show this. So as I said, such complicated stuff is always in mock. So we have an expanded form. So for us, we used it from glycerate 3-phosphate, remove water, this water, and harvest the ATP and form your pyruvate straight. But now for this point here, we have a detailed structure where we have magnesium ion activator and an enzyme there, enolase enzyme. So magnesium ion activator and the enolase enzyme are going to convert glycerate 3-phosphate to N-pyruvate. As you convert it to N-pyruvate, you convert it to PEP. We find this PEP in photosynthesis for C4 plants. So this PEP is going to be converted into N-pyruvate. How? We remove our ATP, and this one is dephosphorylation. So PEP is going to be dephosphorylated and will form N-pyruvate. Now N-pyruvate, as we remove water from N-pyruvate, we are going to be removing that eno group and we form pyruvic acid. So this is it, and that's what happens here. That's what happens around here. Just this step. So I'm going to move on to my fatty acids. So what if I convert that fat to the fatty acid? What happens to the fatty acid? So what happens to the fatty acids? Fatty acids are very long chain organic compounds. So you may find the fatty acids of 18 carbon atoms. What do? These fatty acids are going to undergo a series of breakdown. We break 2 carbon units. So if there are 18, so 1, 2, let me see, I'm having C, like that. So what we do, we start breaking down 2, and every 2 carbon atom unit is going to be converted into acetyl-CoA. You remember that acetyl-CoA is a 2 carbon molecule. It's a 2 carbon molecule. So we are going to be removing 2 carbon atoms from it. As we remove those 2, we form, so this one is going to form its own acetyl-CoA, this one forms acetyl-CoA, that one, and that one, until the whole fatty acid is broken down. So that is it. Then that is it for the fat. So as we break down glycerol, we break down the fatty acid. Then I'm going to move on to the protein. What if I'm having a protein here? What happens to the protein? What happens is that this protein, usually it's broken down. We know this. It's broken down to its amino acid. And after breaking it down to amino acids have very many alternatives. An amino acid can be converted to a pyruvic acid or pyruvate. It can be converted to acetyl-CoA, or it can be taken directly into the mitochondria, and specifically in the Krebs cycle, like that. And we are saying that this reaction is reversible. The only non-reversible reaction we are seeing here is this conversion from pyruvic acid to acetyl-CoA. So these reactions are reversible. What does it mean? It means that when a certain amino acid is in a deficient state, or it's deficient, the body is lacking certain amino acid, we can process our sugar into pigar, then we form pyruvic acid, then from pyruvic acid we form that amino acid there. Are you seeing that sequence? So let me bring it out like this. So if maybe I'm having a certain protein amino acid in deficient state, we can start from our carbohydrate to pigar, then from pigar to pyruvic acid, and from pyruvic acid we form the amino acid. Oh, if it wants small modification, we go to acetyl-CoA, and from acetyl-CoA we can always form that amino acid. Then we can even wait until this acetyl-CoA enters the mitochondria and into the Krebs cycle, and we pick one molecule from there, maybe succinic acid or alpha-ketoglutarate, malate, oxaloacetic acid, or succinate. We get one of them and convert it back to the amino acid that we want. Are you seeing that? Oh, we can even get this fat, turn it to a fatty acid, break down the fatty acid to acetyl-CoA, and we turn the acetyl-CoA into the amino acid that we want. So that's why I called it an interconversion. It's an interconversion of all these three. All these three conversions are happening within the same region. So that is it. This is it. Then I move on to our last part. Control respiration. Remember, our body has to have homeostatic controls on every process that happens within it. And for respiration, we have our control respiration here. So in our control, I'm having glucose. This entire process is glycolysis. No, this part here is glycolysis. Then we enter Krebs, then electron transport system. But in our glycolysis, I've summarized it into from glucose to fructose 6-phosphate to fructose 6-phosphate. We remember this, it was from glucose to glucose 6-phosphate to fructose 6-phosphate, then fructose 6-phosphate turns to fructose 1,6-bisphosphate. But this fructose 1,6-bisphosphate comes from the enzyme. The enzyme that is going to catalyze it is fructose phosphofructokinase. I hope you remember this enzyme. So this phosphofructokinase enzyme is going to be doing the work. Now, so all our control is centered on that phosphofructokinase enzyme, all the control. So this phosphofructokinase is going to catalyze the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Now as we convert it to that, it's going to break down to pigal and dihydroacetone. Then we follow that series of reactions in glycolysis, and we end up with pyruvate. Now as we end up with pyruvate, this pyruvate is going to be converted to our Krebs cycle, converted into the Krebs cycle within the mitochondria. Now, in our mitochondria, we have production of citrate. This is the citric acid compound that we formed previously. It's a 6-carbon compound that we formed just after adding acetyl-CoA with oxaloacetic acid. We formed the citrate. And now, as the concentration of this citrate increases, this citrate is going to diffuse out of the mitochondria. As it diffuses out of the mitochondria, it's going to go within the cytoplasm and look for wherever there is phosphofructokinase. So wherever phosphofructokinase is, our citrate is going to go and bind up with that phosphofructokinase enzyme. As it binds up to that enzyme, it's going to cause hindrance or inhibition. This one is an example, one of the examples of end product inhibition. So we are saying that our citrate is going to inhibit the progress of glycolysis. So it's inhibiting glycolysis. Now, as we inhibit glycolysis, there is no more glycolysis going to occur, no more pyruvate, so no more Krebs cycle. Are you seeing that? So the citrate concentration will go on decreasing like that. Now, We move on to the electron transport system. As citrate concentration is increasing, we are saying that even the electron transport system is going to be moving on and increasing the concentration of ATP. Now, the concentration of ATP increases to the extent that ATP will diffuse out of the mitochondria and attack this phosphofructokinase, the very enzyme that is being attacked by the citrate is going to be attacked by ATP, and it's going to cause inhibition. It's going to cause inhibition of our glycolysis. So that process of glycolysis is going to be inhibited by two end products, ATP and citrate. So it's not until ATP concentration decreases as ATP is being broken down. So as we break down ATP, into ADP and AMP, so we break down this ATP into ADP and AMP. So it's this AMP that is going to go and bind on the phosphofructokinase. So phosphofructokinase has very many receptors and active sites for ATP, for citrate, and for AMP. So this very AMP is going to go to this phosphor to this phosphofructokinase. As it goes to our phosphofructokinase, it's going to activate it. So it's going to activate. Now as it activates, glycolysis moves on. So glycolysis will move on now. So that is what happens. So this is going to be responsible for activating glycolysis, while the two, citrate and ATP, are deactivating it. So what's this? This one is adenosine mononucleotide phosphate. It has one phosphate. So that is what occurs. Go and read using at least this recording. Is going to help you. Yeah. So whatever we've summarized is here. So let me see what we've covered. We've covered glycolysis, Krebs, electron transport system, phosphorylations, plant and animal fermentation, control respiration was the last, metabolism of other substrate. That one is what I called our interconversions of those molecules there. So that is it. So assignment I'm going to leave you with an assignment, and our assignment is going to be or research work. So the research work is going to be look for the differences between aerobic and anaerobic respiration, aerobic and fermentation. Then look for their similarities. After, look for, this one is very important, look for respiratory quotients. Look for respiratory quotients there. So this is the end.